So let's welcome Dr. B.V. Ravichandra to the virtual stage for conducting the session on navigating NDSRIs, challenges, toxicology, and the path to safety.
Thank you so much. Okay. Firstly, thank you so much, Guneet and team and Eminence Group for inviting me to this very important conclave, which is on the focusing on the impurities, and I'll be covering mainly on the nitrosamines and the NDSRI, which is the topic of our now. So everyone in the pharmaceutical is going through this phase where we have to address a lot of challenges from the different regulatory agencies, the requirements changing every now and then. So it is the landscape is keep on changing, like every day, every month. We are hearing the new papers, new updates from different agencies, and the list being updated every day. So what I am going to cover today is very high level and to some extent to the deeper level of the how the safety assessment and how to navigate when you exceed or when you cross these nitrosamines or NDSRIs, especially in your pharmaceutical drug product. So this is a high level agenda, like I can say the items which I'll be covering. I'll be covering on a very high level how FDA and then the EMA and then the acceptable limit looks like, and how the in silico tools we can use to address these nitrosamines or NDSRIs from the mainly from the safety point of view. And from the experimentation side, I'll be touching upon the EAT, the Enhanced Ames test, which is used for the evaluating the in vitro mutagenicity, and then the second mutagenicity assay, in vitro metabolism, and then the last in vivo mutagenicity, which is the last resort to address this when you exceed the AI limits. And again, how we can explore the emerging technologies for these mutagenicity evaluation. Also, I'll be covering to a certain extent. So I would like to apologize. Maybe in the beginning itself, like I'll not be covering anything on the basics. I assume everyone here is well aware of the nitrosamines and then the what is CPCA, what is AI, and I'll not be covering anything on the basics because I wanted to make use of this time for the actual content on the topic instead of covering and wasting a little bit of time on the basics. So if anything you would require, maybe you can just Google it. I think everyone might be aware already, so I don't need to touch upon those things. So I'll be directly touching on to these actual content. Okay, so this is the basic slide which I can say on the safety assessment. Being a toxicologist in a pharmaceutical industry, I typically get a request from R&D scientist, like either it can be from analytical or a formulation scientist or even a regulatory guy. When they exceed the CPCA limit for a drug product, which is either listed in the FDA or not listed in the FDA, or mainly I'll be talking about FDA, but it's more of a regulatory agencies, or you can consider all the agencies because I work more with FDA, so I tend to take that word more often. So when we cross these limits, right, so they come to us and then asking for the relaxation of these limits. And currently, the FDA and EMA list down a lot of approaches how we can address when you exceed these CPCA limits in any drug product or API. Mainly based on the compound specific data. When you don't have any compound specific data, right? So you can go with read across approach, toxicological read across approach using the surrogates with the reported AI or toxicological data, whichever available for that particular read across molecule. I'll be covering this to little extent, and then conducting these experimental studies, which are currently listed by the FDA and even EMA to some extent. But FDA is more focused on these second in vitro mammalian assay. EMA doesn't mandate that, right? So EAT is good enough for the EMA, but FDA requires multiple studies to control a limit of 1500 nanogram. And the results again depends like what results you're getting in these preliminary in vitro results covering EAT, in vitro mammalian cell mutation assay, and in vitro metabolism. Depending on the result again, if it comes negative, you can control again up to 1500 nanogram for FDA and both EMA. And if it comes positive, the last option is either you control as per the CPCA limit, or you go ahead and conduct the in vivo mutagenicity, which is the last resort. But again, that's not everyone's a choice to go ahead and do this in vivo mutagenicity because of a very expensive, this is a very expensive study and then the time consuming study. So once you do these in vivo mutagenicity, again, depending on the result, based on the positive or negative, you can control this limit as per the CPCA if it comes positive, or you can even negotiate with the agency doing the BMD approach and then deriving a certain limit. And then if it is negative, definitely you can go beyond 1500. EMA gives that provision to control as per the non-mutagenic impurity limits, QT limits, but FDA currently is not allowing to control, in spite of proving your compound a negative in vivo mutagenicity. You cannot control to ICH QT. You have to control it as per your current trend data. But again, definitely more than 1500. But again, they don't give to what extent you are allowed to control those limits. Okay, so before I move ahead, I'll just give you how FDA is currently listing on the database, which everyone might be aware. The FDA nitrosamine web page that lists all these nitrosamines with the limits. Currently, there are 284. There could be some changes to this number. I updated this, I think, a week ago. Uh, uh, so roughly 284 NDSRIs currently listed, and this is the breakup of the different CPCA categories, how they are uh, broken down like 1 to 5. So majorly it is falling in category 4 and 5, which have a limit of 1500 nanogram per day. Okay, so how these FDA and EMA again compare the A limits? When I look into the EMA and also the FDA limits for both same nitrosamines, uh, nowhere FDA is having a more relaxed limit as compared to EMA. Practically, there are only three limit. Maybe I'll just go to this slide where AI published by FDA is more as compared to EMA, but it is practically not the case because EMA considers a lower limit as compared to FDA because of 18 and 26.5. That makes the difference. Otherwise, there is not much difference here. So EMA gives more often the IRA limit as compared to FDA, and they are more flexible in accepting these relaxed limits. And they also published on their database as well. So this is how it lists down the lot of NDSRIs where EMA has considered already a non-mutagenic impurity as compared to FDA, which is controlling at a tighter limit of 1500 nanogram per day. That's again very challenging for some of us to address both FDA and EMA. So there are certain interim IRA limits again published by AI. They are not applicable only till August of this year. Okay, so coming to the read across. So read across is one of the vastly used methodology in the toxicological risk assessment to go beyond the CPCA limit, which is the limit which is set by any agencies. Uh, Currently, FDA list of these five molecules, or I can say it includes small nitrosamines, which can be used for toxicological read across. So I don't know, means if anyone tried other than these five, but again, we are trying, but again, we are not hearing any feedback from the agencies. But these are the five molecules listed by FDA, which says you have to use this, or you can use also the other surrogates with a proper justification, which I have personally done, going beyond these five and relaxing more than the CPCA limits. And there are certain NDSRIs limits also published by FDA using these read across, but none of them crossing 1500 nanogram. That's again interesting to note. So, okay. So touching upon what I was covering earlier, the read across, and then the For read across again, we do use in silico QSR tools. Mainly, these are the three commercial uh, OECD validated softwares currently FDA is accepting. And the Case Ultra, which is provided by Multicase, Direct Sara by Nexus, LASA, and then the Lead Scope by Instem. So they have both knowledge based and statistical based complementary tools, and they are validated as per the OECD. Currently, these are accepted, and Nexus also gives you a CPCA limit, and any one of these is acceptable by the agency. You can make use of these QSR tools for the read across. Many training sets these softwares gives you. You can certainly use them, and they build on the hypothesis and go for read across as well. Okay. So coming to the experimentation, means toxicology studies. What currently FDA list is the four studies. One is the EAT, enhanced aims test. You might have heard a lot of times this EAT. On September 2024, last year, FDA came up with the additional requirement of an in vitro mammalian cell mutation assay. Again, they're not specifying which assay. They're broadly saying it as in vitro mammalian cell mutation assay. And then the third is in vitro metabolism data, which they also mention, including the human hepatocytes and the microsomes. And the last one is in vivo mutagenicity when you want to go beyond 1500 or any one of these first three assays comes positive, then you have to conduct this in vivo mutagenicity. The last but not least is I not covered here any carcinogenicity studies, which is the ultimate study. But again, that's again not everyone's choice to do the carcinogenicity because they are all lifetime studies. It may take more than 2-3 years. So when we dig internally at our end, also like what is that second assay which we can do, right? Again, they mentioned the OECD guidelines, but there are we found out like there could be these two assays, in vitro TK gene mutation assay, which can be done in human or a mouse cell. Currently, the OECD guideline says that, and the in vitro HPRT gene mutation assay, either the human or a mouse or a hamster, right? But these are for the normal impurities or the normal any compound. But what about for the nitrosamines? And we deep dive again into the literatures. So what we found, these could be these are the this could be the possible second mammalian mutagenicity assay because FDA is not clear what assay they want. But if I conduct as per the OECD either of these two, again they may say that may not be valid. So when we deep dived into again the literatures, so FDA published 3 to 4 publications recently as well as one publication, and there are a lot of FDA workshop going on. So we attended that as well. There are a lot of industry expert and consultants we reached out, right? So where we found out like these publications gives hint to which assay you can be going to in vitro TK gene mutation assay using the human cells. Because again, we had a confusion in the human and mouse, so but it hinted towards the human cells. And then the in vitro, again, HPRT gene mutation, again, these publications hinted towards the human cells. There are multiple other assays listed on here. I don't want to read out, but again, the acceptability is again questionable. But currently, these are the requirements which agency is asking for. You can opt any one of these assays because as they are not specifying anything, I think you can still fight with them. So maybe as a strategical approach, you may take up as a phase-wise approach. One maybe like you can start with a non-GLP EAT assay. Depending on the result of positive or negative, you can take up the GLP phase 2, where you can cover all these three assays: EAT, GLP, mammalian mutation assay, either human or any other cells, or the in vitro metabolism using human hepatocytes or microsomes. And then there's a phase 3, depending on the result, as I said, you can go to TGR, in vivo mutagenicity assays. Also, there are a lot of technologies like duplex sequencing and I5 sequencing. Again, currently, because of certain limitations and in the lack of validation, this is still in a developmental stage. Okay, so just I would like to cover like what is AIMS and what is enhanced AIMS test, just for the sake of understanding. So As everyone knows, this AIMS is the gold standard for the mutagenicity evaluation, which is currently in use since many decades, right? So typically, it is done in 5 tester strains with both uh, minus S9 and plus S9. Uh, those who are here, non-toxicologists who are not aware of this, what is F9? This is something like a liver fraction which helps in the metabolism. So it mimics the liver. So minus S9 is without that liver fraction, and plus S9 is for with the liver fraction. So that mimics the metabolism which is happening in the body. So there are two methodologies which we used to follow in names: plate incorporation and pre-incubation. When I say pre-incubation, it was for 20 minutes, but the recent enhanced AIMS test protocols, again, more or less the same, but except a few things are changed, which are highlighted here. So addition of hamster resident, which is up to 30%, and then the addition of the positive controls. Apart from the routine positive controls, you have to include uh, a small nitrosamine and an NDSRI positive controls, and the pre-incubation time again extending up to 30 minutes. So there could be multiple changes to these enhanced AIMS tests, which are already there, which many people have started using it. Again, the terminologies earlier it was only AT AIMS test. Now it has become EAT, and later it may become again the modified AIMS test. Right. Okay, so the second mutation assay, which I was telling you before, like you can even opt like PKM gene mutation assay or HPRT gene mutation assay. And then the supportive metabolism studies, as I said, you can make use of primary human hepatocytes or the microsomes for these in vitro metabolism data. But how much extent the data is required to support your mutagenicity assays, no one is very clear. And we are also in touch with FDA and other regulatory agencies what data is required. But some people goes up to identifying certain metabolites, even profiling it. But none of us are very clear what exactly is required. Okay, so when you get any AIMS positive result, these are the studies which ICH-M7 list, and which every one of you might have seen when you have gone through the ICH-M7. The topmost assay which is listed here is the again the gold standard I can consider from the in vivo mutagenicity, which is TGR, transgenic rodent mutation assays. And there are multiple other assays listed down in the ICH-M7, but currently for nitrosamines, TGR mutation assay is the one which is accepted. Okay. So before I get into actual how these mutations and occurs, this is one of the very nice slide presented by Pfizer in one of the US FDA workshop. I took it, and this uh, covers the adverse outcome pathway. If you break it down into mutagenicity and carcinogenicity, because mutagenicity is just an event. Okay, it is not an adverse effect, or like I can say, which is visible. Carcinogenicity is something which is apparent and visible, because mutagenicity is just an event. Because day in and out, we are mean within the human body, a lot of mutation occurs which gets repaired on a normal basis, because our cell system is so strong and robust. They repair those mutations and then correct, but that doesn't lead to cancer, right? So, but again, these nitrosamines are going to cause a mutation which is going to fix and then turn into a cancer, carcinogenicity. The carcinogenicity is an actual outcome, and mutagenicity is just an event. Proving that your compound is just a non-mutagenic, it doesn't mean that your compound is non-carcinogenic sometimes, right? And vice versa also is possible. All carcinogens are non-mutagens, means are not mutagens. So that's also there. So these are the possible assays which we can make use of. This is typically the how nitrosamine compound or any typical DNA reacting compound reacts with the DNA, and this is mainly for the nitrosamine, how the alkylation of DNA happens, and then how insufficient or incorrect DNA repair results into demutations and resulting in further into the cancer, which is the carcinogenicity. So again, I bifurcated which are all the assays which covers the mutagenicity, and then which are all the assays which covers the carcinogenicity. Carcinogenicity, obviously, it is only the rodent carcinogenicity, which runs for lifetime in either rat and mice. Mutagenicity, as I said, the transgenic reader mutation assay in vivo, Big Blue and Mutamouse, they're routinely conducted. Again, the Big Blue is the one which is mainly done. And there are a lot of other DNA damage identification assays like Comet assays, and then the UDS assays. But again, they're all currently in developmental stage. I don't know how agency is going to accept those things. Typically, whatever I outlined in the previous slides, they are the one which to be done. Okay. So as I was explaining you, this is a Q&A statement which I picked up. This is very important statement. If you read out the highlighted part here, a in vivo mutagenicity assay alone are currently not validated to directly assess the cancer risk because of the reason which I mentioned. Because mutagenicity is only an event, cancer is an outcome, and because of that, endpoint is a mutation and not carcinogenicity. Okay. Okay, so as I said, again, all mutagens are not carcinogens and all carcinogens are not mutagens. This is very obvious. Again, why agency? Uh, Again, certain things are like there. Why agency is not accepting the LTL approach for the nitrosamines, because there are reports. If you look at even this publication 1991, not the recent one also, single dose even causes the cancer. So such is the impact of these nitrosamines. So that is why nobody is accepting now the LTL approach uh, for the nitrosamines because of such risk. Okay, so coming to the last experimentation part, which is the transgenic rodent mutation assay, which is in vivo covered under in vivo mutagenicity assays. There are two major assays. One is the Big Blue, which can be done in rat or mice, and the Mutamouse, which can be done in mice. So the most maybe I can say commonly done assay is the Big Blue in rat or mice, depending on your requirements. So I'll be just covering what exactly is this transgenic rodent mutation assay. You might have heard Big Blue and Mutamouse from any of your toxicologists in your organization. So Mutamouse is again a Japanese CRO developed assay. They again they uses the transgenic animal models to investigate these mutagenicities. And then the Big Blue, again, there are very limited CROs who can do these Big Blue assays. There are hardly 2, 3 CROs globally they can do. That is why the timelines and the slot availability is a challenge, and they are very expensive. And the test item requirement is again huge. It can go up to 200 to 250 grams. So you can imagine, like for nitrosamine to get such a scale, means the amount of impurity is really tough. Both these transgenic rodents means mutation assays are currently accepted as per the OECD 488 guideline, and everybody is doing these assays. Even in EMA, recently considered a couple of these NDSRIs as non-mutagenic impurities based on these in vivo mutagenicity data. Either you can get the BMD, which we call benchmark dose, or a no-gel, no observed genotoxic effect level in these assays, which can be used to set a limit. So this is a Big Blue assay. As I said, it can be done in both mice and rat. I don't want to get into all these minute details, but typically how it runs, you dose the animals, and then you collect the tissues, and then you isolate the DNA, and then you extract and then infect these into bacteriophage viruses, and then again incorporate these into the bacteria, and then you grow how it is seen in typical Ames colonies. Similar way you also see here the colonies. So what we are trying to do here is you're administering the compound to induce mutations, and you're allowing the time to fix, and then you are making that to phenotypically express through the microorganisms, which is seen as the colony counts. So the genotypic whatever the fixations happened, we made it to visualize as a phenotypic expression. That's the typical uh, principle of this assay. And typically, there are like four tissues which we need to collect here. One is the site of contact. When you talk about the oral product, it is typically a duodenum or a stomach, which is the site of contact. And then this systemic exposure, because liver is the first organ when you take any product orally after the GI. So the liver is also the site of metabolism. So that is the most preferred organ. And again, you need to cover a rapidly dividing, which we already covered, which is duodenum, and then the slow dividing, again the liver. So you can opt a liver or a duodenum as the minimum organs. Apart from this, you can go for other tissues as well. People do collect testes and keep it. Okay. So there are other. That's all about the in vivo assays which I wanted to cover, and then there are a lot of other technologies, new technologies currently available. But again, there are a couple of CROs which started and now they shut down, and there are new CROs also working on these error-corrected new generation sequencing. Typically, what we have seen here, genotype to phenotype expression. But these technologies will help you to identify the genome sequencing, like what is the mutation happening in that particular gene. So directly you can see that through the sequencing uh, technology platforms. So the one table which I listed, shown on the right side, that gives you the how the evolution happened with respect to the sequencing. Currently, we are at a stage where this duplex sequencing is there. So when you sequence these millions and billions of nucleotides, there is a chance of error. So because again, you don't know these errors are really because of error happened in the platform, or is it a mutation. So for that reason, it was not extensively used earlier, but there are a lot of new technologies which have come up now, duplex sequencing, that reduces the error significantly. That is why this is called as ECNGS. So error corrected NGS. What they do here, uh, earlier they used to do the single strand sequencing, but now they are doing the sequencing for both the strands, and then they are matching. So the error chances goes down significantly. If you see 5 into 10 raised to 8, so that is the error frequency, that such is a low. So I don't want to get into these how these ECNGS and all done. Okay, so there is another sequencing which is hi-fi sequencing. Currently, also many people have exploring these similar to duplex sequencing, but the principally it is little different. So currently, this is offered by Pacific Biosciences. FDA also working on these next generation sequencing using all these platforms. Earlier, FDA collaborated with one of the CRO, but that is currently shut down, which is Twin Biostrand. And now there are other CROs working on these duplex sequencings. Okay. So, just to give you one of the latest paper where they compared these error-corrected duplex sequencing with the in vivo mutagenicity and then the other assays, where they dosed rats for 28 days, which is the big blue, and they also done this duplex sequencing and comet assay just to show how these assays compare. And they dosed here the known positive nitrosamine, which is the NDEA, right? And surprisingly, they found out duplex sequencing is more sensitive assay as compared to the big blue and then the comet assay. And this paper also concluded, right, that the duplex sequencing and comet can be a better alternative also to these the very expensive and time consuming. Usually duplex sequencing are not so time consuming. They are very easily doable if they are robustly developed. But again, that's the challenge which everyone is facing now at the CRO end. So that concludes my presentation, just to cover on the what could be our future of these nitrosamines. As I said, there could be multiple changes to EAT methodologies, which may make it like modified EAT, and then We don't know how this TGR, because everyone can't do the TGR, which is very time consuming and very expensive. So we don't know what is the feature for that in vivo mutagenicity, how we can go beyond 1500 nanograms, mainly for the FDA, because EMA already considered this as an NMI whenever we show it as negative. And then there could be multiple alternative in vitro assays. Currently, HESI is currently working on four different approaches. If you look at the picture shown here, there are a lot of groups which are working on the even the QSR and in the QM. So that's again the one of the latest concept. It's called as quantum mechanics. People are using it. A lot of publications around it, and Jacob Coastal is currently leading that group, and they are working extensively on how to address these interactions. And then there are a lot of in vitro approaches, as I said, there are a lot of new assays may going to come up in future, and definitely these assays would replace the TGR, which is the time consuming. There could be a lot of advancement in the QSR, which already is happening, because earlier QSRs and now the current QSRs, there are a lot of changes, and they incorporated a lot of NDSRIs and PPCA AI limits into those assay. So there could be a lot of developments from the ICH and the OECD side as well. And again, we don't know like how these NDSRIs would be going ahead. Again, we may call it in some near future that could be called as a non-COC, mainly the NDSRIs I'm talking about. But there's a long way to go again. Uh, so that concludes my presentation. Thank you so much, everyone. If you have any questions, I'm ready to take.
All right. Thank you so much, Dr. B.V. Ravichandra, for that phenomenal session. And that really helped everyone to navigate through NDSRIs. And now we will move on to the question and answer session. So let's see what's the first question. Okay, so it's from Srinivasa Aghuram M from Heterodux. Now, the question is, what criteria determine the necessity of vivo follow-up assays for ME's positive nitrosamines?
Okay, what criteria determine the necessity of in vivo follow-up assay for Ames positive nitrosamines? Okay, so it all depends on you, because again, if you need anything beyond 1500 nanogram or the PCA limit, which is set currently by any of the agencies, if you want anything beyond, so just you want to prove it as a non-mutagenic in any of the in vivo assays, possibly. So then that's your choice. It's not mandated by anyone. There is no as such criteria for it. But if you got a positive result in any of the in vitro assays, definitely the in vivo is the next step, as I said. But again, it all depends on what limit you want in your product. Hope that addresses your question.
Yes, yes. I mean, that was. We also have a new question. I will be putting that question on stage. Perfect. So this is the next question actually. But we've got this question from Mr. Prashant from Cohen's. Can you hear
Okay.
me? Yeah.
So which bacteriophages are being used, and are they selective to different nitrosamine impurities? Bacteriophages. Think I'm not very clear. So in my whole presentation, I mentioned only one place, the bacteriophage, which is used in Big Blue. So that is not going to be different. I think, I don't know, the question is not very clear.
I
But
will
if
do
you're
one.
asking,
In
sorry,
that case, I will ask Prashant to come on screen. Prashant, can you opt for the raise hand option? If Prashant can listen to me, Prashant, can you just raise your hand so that I can invite you on stage? You'll have an option of raising hand. Can you do that, please? No, I can. Prashant, you.
No problem. I think if he's asking from the mainly from the EAT side, if he's confused about the what bacterial strains to be used, so it is not different for any of the like it is not different for the different nitrosomes. It is typically the five test strains. If he's asking a question on the what bacterial strains to be taken for EAT, so that is the answer. That's my answer. But if he's asking the question on the big blue, uh, it is going to be a same bacteriophage. That's not going to be different for any of the different nitrosomites.
No problem. Perfect. Dr. Ravi, in case if Prashant has any follow-up question, I can maybe connect with him offline and
Sure.
get that clarity. Okay. Yes, I think Prashant has mentioned. Yeah, we can go ahead with this question for now. Uh,
Okay.
this is something which we had
Okay.
sent it.
Reference guide to latest regulatory updates. How should companies interpret and implement them? Reference guide is the FDA side or the EMA sites or the Health Canada side, whichever the agency you're currently working for. They're the main source where the latest updates you can take it. And there are a lot of new publications coming up, mainly by these AC group, and then even FDA is publishing. There are a lot of, if you see, I mentioned also the Lee et al. 2023 and 2024. They are also published by FDA people. So a lot of FDA work is going on. They're publishing continuously. But at the end of the day, you have to refer back to your guidelines or the web page for any of these updates.
All right. So yes, this question is all right. Also, we have. I think Prashant has mentioned in the chat box, sir. So you have, I mean, answered his question.
Okay, great. Thank you.
Yes. Where's the question? Okay. The next, there's one more question. Recommended as per AI, FDA can consider as final AI for
Okay.
NDSRIs.
Recommended AI as per FDA can be considered as final AI for NDSRIs. Yes, you can consider. That's how currently the practice is.
Okay. All right.
Is this question answered? Because this is put in the chat box. So it's very important, guys. It's very difficult for us to manage the questions from the chat box. If you put it in the Q&A tab only, that will be easier. We were almost going to miss the session, miss the question by Dr. Reddy from Wanbury. So is that question answered? Can you please
Yes.
tell us? Can you show a thumbs up,
He did mention that.
Dr.
Yeah.
Reddy? Okay, he has mentioned. Thank you. Okay, all right.
So I Yes, so I think we don't have any more questions now. In case if we have any questions, people please put it in the Q&A tab. And I think most of your questions have been answered. I think Dheeraj will be taking forward.
Yes. So once again, thanks a lot, Dr. B.V. Ravichandra, for this session and answering all the questions. And I'm sure there will be more questions which we can, always connect with you later on. And now it's time we move to our next session. And our next session is about
Thank you.
group activity with. Okay, just one sec, sir. We have one last question before we move ahead. Is it only for US? So it is a continuation of the previous question.
It is for US only. Means maybe if you can elaborate the question, please.
Let Let me invite Miss.
Yeah, maybe you can invite Dr. Praveen as well.
Yeah, okay. So this question is by, okay, it's by Praveen. Can Praveen please join the stage? Can you invite
Yes,
him?
invite, yes.
Thank you. Praveen, you're on mute if you're trying to ask a
Yes,
question. Can
yes.
you?
Am I audible?
Okay.
Yes, we
Yes.
can hear you.
Yes. See, when FDA publishes the AI acceptable intake limits for nitrosamine, it is only for the US region. Okay, so the EMA
Correct.
will be having different AI limits for European region because some of the cases, okay, so FDA publishes like 26 nanograms is the maximum acceptable limit for one of the nitrosamine impurity. But for same nitrosamine impurity, EMA says that it is 80 nanogram. So in that case, only for US, you can go ahead with 26 nanogram. But for Europe, you have to go ahead with 18 nanogram. If you want to have both regions same limit, you have to go ahead with 18 nanogram.
Correct. The stringent limit you can adopt, as I
Yes,
mentioned earlier also in the slide. So it's the difference between 18, for which means class one category. So
yes.
obviously, if you're working globally for the catering to different markets, certainly you can adopt to the lowest limit, which is there in the amongst the different regulatory agencies. In many of the
Yeah.
cases, for class one, definitely EMA would be the case because 18 nanogram is much more stringent than 26. But many of the other cases, FDA is little more stringent as compared to EMA. Yeah.
Whenever the regulatory authorities, any regulatory authorities publishes the results or the guidelines, that is specifically their regions, you can use this as a reference to convince the other authorities.
Yeah, certainly you can convince, but again,
Yes,
they will listen because they also gave an option. You can
yes, yes,
justify
yes. You're right.
the limits. You can certainly do a control correspondence for our product. They'll just certainly answer. But they did also mention FDA, if you look at the web page, that they do differ with the other regulatory agencies, which typically they are mentioning for EMA in consideration of certain nitrosamines on NDSRIs, because EMA considered many of these nitrosamines as NMIs, but FDA is not considering that. So they did mention that they may differ with the other regulatory agencies, but certainly you can try with your justifications.
All right. Thanks a lot for concluding. One last question from Dr. Praveen and taking it up. We will now move ahead on to our next session. And once again, we have a claps on the emoticon.